Welcome back to Introduction to Genetics and Evolution. In the previous videos, we talked about the process of recombination and specifically the sub-process of crossing over. We talked about using the fraction of recombinant offspring to figure out how far apart genes were. In this video, we'll talk about how to leverage that calculation to generate a map using 3 or more genes, and basically to figure out the order genes are along a chromosome without having to get its full DNA sequence. So, what if you have 3 genes? Let's say that you have 3 genes A, B, and C. And let's say you know the phase of these genes where all the capitals came from one parent, all the lower cases came from another parent. You have a heterozygote. So it's big A, little a, big B, little b, big C, little c. But in this phase as depicted here, and you're crossing it to the little a, little b, little c, just like what we did in the previous video as a test cross. Okay? Now you know the parental phase is A, B, C, capitals, or all lower cases. But what is the linear order? Now what you don't know is this the order? Is B between A and C? Or is it this order where A is between B and C? Or is it this order where C is between A and B? The answer is you don't know. So this is what we're going to try to calculate. So let's look at this. Which of these is parental? Which of the set of possible offspring is parental? Now, these top 2 individuals are definitely parental, right? Because they have the full combination, capital A, capital B, capital C, and lowercase a, lowercase b, lowercase c. These other ones all have some sort of recombination event that's happened in there. We don't know the details. Let's put some numbers in there to see what happened. Well, here are some numbers. Maybe you scored either molecular markers or phenotypic markers, and you get these sorts of numbers here. Now, the top 2, the parental types, are always going to be the most abundant. You will nearly always see that. At worst, they'll be equal to the others, but more typically, they will be more abundant if you're looking at genes along the same chromosome. We have a bunch of much rarer types going down. Now, what I'd like to do is I'd like to dissect the problem. Rather than trying to look at A, B, and C simultaneously, let's just ignore C for a minute and just look at A and B. Okay? So just looking at A and B, we can identify the top 2 are parental again. They have big A and big B together, little a and little b together, which matches the phases found in the original parent. The next 2 are recombinant because you have a big A with a little b and little a with big B. The next one is actually parental. This is important. This next one is parental-like. It's still big A, big B, and little b, even though we know the C is out of phase with the others with respect to the parents. But again, we're breaking down the problem just to look at A and B. Okay? And the bottom one here is recombinant. Big A, little b, and little a and big B are together. So when we add these up, what fraction are recombinant? Well, it's 15 plus 13 plus 1 plus 1. So that adds up to 30. That's up to a total of 30. The total here is 1,000, if you add up all those numbers together. So our recombination fraction in this case would be 3%, or 0.03. Okay, so that is the recombination distance between A and B. Okay, so we calculate that. That's between A and B. 3 centimorgans apart, or 0.03 recombination fraction. What about between A and C and between B and C? Well, we can repeat the same process. Let's go ahead and do that. Between A and C, which ones are recombinant? This would be recombinant between A and C. This would be recombinant between A and C. This would be recombinant, and this would be recombinant between A and C. The bottom one would actually be parental for A and C because you see the capital A and capital C are together, or lowercase a and lowercase c are together. So between A and C, we have 18 and 13 is 31, 31 and 15 is 46. 46 over 1,000. Okay? What about between B and C? Which ones are recombinant, which ones are parental? I'll change the color of my diagram here. Between B and C, this would be parental, this would be parental, this would be recombinant, this would be recombinant, because again we have big B, little b, big C. This would be recombinant, this would be recombinant. So in this case, we'd have 20 over 1,000 will be recombinant between B and C. So let's put all these numbers together. There we go. So we have, between A and B, we have 3 centimorgans. A and C, we have 4.6 centimorgans. B and C, we have 2 centimorgans. So we can, from this, figure out the linear order of genes, right? What should happen is the distance from one end to the middle plus from the middle to the other end should be about the same as the total distance. Imagine that you're going from, say, New York to Florida. So you have 3 points, New York, Washington, D.C., and Florida. If you add the distance from New York to Washington, D.C., and the distance from Washington, D.C. to Florida, that should be about the same as the distance from New York to Florida. So in this case, we could do that, and we can say, because of this, A and C must be on the outside. So you have to have A at one end, C at the other end, and therefore necessarily B will be in the middle. So this would be the correct order, right? Because here we have distance from A to B is about 3, distance from B to C is about 2. The total of the two is close to 5. It would round up to 5. So now you may be wondering, why don't AB plus BC? Why don't those two distances add up exactly right? Why doesn't it add up exactly to 4.6? Well, the reason is something that you actually noticed in when we were calculating these numbers, is that we have two things that we're calling parental, but they're actually recombinant, right? That here at the bottom two, this individual and this individual, we were counting as parental for A and C. But in fact, they're double crossovers. So not only should they be counted, but they should be counted twice. But we didn't do that. So this is why it doesn't quite add up exactly. We'll come back to this in just a second, but first let me emphasize a different point. When you're trying to figure out the linear order of genes, there's a few tricks you can use. First, if all the loci are linked at some level, and I don't mean completely linked, but I mean that they're on, for example, the same chromosome and reasonably close together, the largest two, the combinations which are the most abundant, so that's just what we saw last time with capital A, capital B, capital C, lowercase a, lowercase b, lowercase c, those will always be the original non-recombinant parentals. So that'll help you determine the phase. The smallest two, basically the numbers that are the least frequent, such as what we saw in the previous slide of capital A, little b, capital C, or lowercase a, big B, little c, those would be the double crossover. Well, why is that? Why is the smallest two the double crossover? Let me go back just to show you that for a second. These down here were the double crossover types. And you see that they were the least abundant among all the offspring. So why is that the case? Well, again, double crossovers, there are basically two things happening together. So in this case, we have the little a, big B, little c as a double crossover gamete. So imagine that you have a 1% chance of recombination between A and B, and a 1% chance of recombination between B and C. Then what would happen is you could actually multiply these two probabilities. So it would be a 0.01% chance of a double crossover. Essentially, you're taking a rare thing and multiplying by the probability of another rare thing to get this double crossover class. They will always be the rarest class from any particular cross. And again, you can identify which marker is in the middle because it'll be that rarest class. The alleles at 2 markers will be parental relative to each other, while the allele at the third will be recombinant. So we could have immediately looked at the previous slide and said, oh, I see that little a, big B, little c is the least frequent category. Therefore, this must be the double recombinant. And if you notice the double recombinant, you immediately know that this one must be the one that's in the middle. So we can leverage this. Here's an example. This is not the same as the previous example, although this adds up to 1,000 as well. Look at this quickly. Which one would be parental? Look at this set of numbers. Which two types would be parental? Well, I said the parental group would be the most abundant group. So clearly in this case, most abundant group is capital A, capital B, capital C, and lowercase a, lowercase b, lowercase c. That establishes your phase already. So we know that the person who we're measuring recombination in looks something like this. What is the rarest combination? What are the rarest two combinations here? In this case, the rarest two would be these two. So what we said is that this would necessarily be the double recombinant class. Given that, when we look at it, A and B are in the same phase relative to the parents, C is in a different phase. A and B are in the same phase relative to the parent, C is in a different phase. So in this case, we can infer the order is actually A, C, B. And then we can calculate the distance between these two genes, or between these pairs of genes. So why don't you go ahead and give that a shot. See if you can calculate the distance between them, and you can look online to see if you got the right answer. What would it mean if you got map distances like this? Let's say between A and B, you got 11% recombination. Between B and C, you got 49% recombination. Between A and C, you got 50% recombination. What would that mean? Well, there's a couple of possibilities here. Now, it could mean that A and B are reasonably close together, and C is really far away on the same chromosome, right? Just because you have a recombination fraction of 50% doesn't necessarily mean they're on different chromosomes. Could also mean they are on different chromosomes, right? So you could have this, where this is on one chromosome, this is on another chromosome. You could have this, or you could even have this other order. Basically, one point I just want to toss out there for you is that above about 40%, map distances are very inaccurate, and maybe basically 50%. So I wouldn't ever trust, when you're determining an order of genes, any map distance you see that's more than 40%, because at that point you really just have no information. So if you have something like this, where A and B is 11%, B and C is 49, or even 43% or something like that, A and C is 50% or even 45%, basically you don't know how far apart these are. You just know that A and B are linked. So all of the information you have in this case is that A and B are nearby, and you have no idea where C is. C could be really far up this way on the chromosome, it could be really far down that way on the chromosome, or it could be on a different chromosome. I just want to emphasize that because some people use these numbers as though they have this absolute precision to them, but we're actually measuring rare events. So when you get something on the order of 49%, or really anything above about 40%, don't have too much faith in that number. Okay? Well, thank you very much for listening, and I hope you found this enjoyable.
Hello, and welcome back to Introduction to Genetics and Evolution. I created this bonus video because some of the students in the class said they'd really like to see a more challenging recombination problem of the type that we discussed, but going further than the examples that you saw in the earlier videos. So let's dive right in. Let me start with a few assumptions. Now, the assumptions we'll be looking at here is that we're scoring offspring from a back cross. This is a key word here. So a back cross is a pairing between an individual that is heterozygous for all three markers that we're looking at to another individual that's homozygous for all three markers. So the offspring from this are called the back cross offspring or back cross progeny. And those are what we're scoring. Now, in scoring those offspring, what we're actually measuring is recombination that occurred in the meiosis of the heterozygous parent. That's what we're actually examining by looking at the genotypes of the offspring. Now, the way we're going to do this is we're going to identify and delete the contributions from the homozygous parent to all the offspring. Now, the homozygous parent may very well have had plenty of recombination. We actually can't tell, since they are homozygous, if they have a recombination event, for example, between the A and B gene. If they're homozygous big A, big B, we can't tell the difference between one big A and the other big A, or one big B and the other big B. So what we'll do is we'll look at the offspring, we'll figure out what the homozygous parent contributed, and just take that out. What that leaves us with is effectively what was in the gametes of that heterozygous parent whose meiosis and recombination we're studying. So the second step then will be to infer the phase of alleles in the heterozygous parent, basically the combination of alleles that were on the same chromosome that came into that parent. This is sometimes referred to as gametic phase as well. The third step will be to infer gene order in the heterozygous parent. Basically, we'll, again, like what happened in the earlier videos, we'll try to figure out which gene was most likely to be in the middle. And the last step will be determining the recombination fractions. So here's a sample problem. If you'd like, feel free to pause the video right now and try this on your own. Otherwise, I'm just going to go ahead and launch in. Okay, so here's the problem. We see that the set of individuals on the left are heterozygous for D. Individuals on the right are homozygous for little d. We see individuals here are big E, big E, big E, little e. We see big F, little f. We see little f, little f. So what do? Well, the first step, as I said, was to identify the contributions from the homozygous parent and delete. Now, how do we figure out what the homozygous parent gave? Well, the homozygous parent is always giving the same alleles. So looking at all these offspring here, so looking at, for example, the D gene, what does every single individual here in the left column and right column have? They have one allele that is shared by all individuals. Which allele is that? Well, that would be the little d. For the E gene, it's the capital E, and for the F gene, it's the little f. So these are clearly the contributions from the homozygous parent. So the homozygous parent, let's just say it was the dad, would have been little d, big E, little f, little f. So we've identified their contributions, so the next step we do is we delete them. So we literally just, boom, they're gone. So now we're looking at basically the eggs of the mom. Okay? So these are the eggs from the mom because we've taken out what the dad put into the offspring. So now what we have here is what was in the eggs, the unfertilized eggs from the mom. So the next thing we do is we try to infer the phase. And again, this is sometimes called gametic phase if you try to Google it or something. The way we identify that is we look at the two most abundant classes, and those are the ones that are in the parental phase. So looking at this, these two are the most abundant. Big D, little e, little f, and little d, big E, big F. So what that suggests is the heterozygous parent had one homologous chromosome with big D, little e, and little f, and the other one with little d, big E, big F. Now I should emphasize that that heterozygous parent thing in the top right there is unordered. We don't know that the order of genes is D, E, F, but we know that that is the phase, that capital D was with little e was with little f, for example. So we've inferred the phase here. Now we don't know the order, so let's figure that out. Well, the next step is to infer the order of the genes. So do you remember how we did that? In the past, the way we did that is we looked at the two least abundant genotypic classes. In this case, it would be these two. Big D, big E, big F, little d, little e, little f. Now what we have to do is, so these are the double crossovers, right? They're the double recombinants, double recombinant individuals. So let's compare those two classes with the parental phase. So we have the parental phase. This is just the same thing from this top right corner brought down. And here's the double recombinant. Now, when we look at that, there should be two alleles that are basically in the same phase as the parent and one that's different, right? So in each of these things. And if you look at the top row, little e and little f are together in the parental and in the double recombinant. D is different. On the bottom side, little d is, we have big E and big F, but we see the D's are not in the same orientation. What that suggests, since this is a double recombinant, D must be in the middle, because in order to go from the parental to the double recombinant, you need two recombination events. Okay? So since D is the oddball, D is the one that is not in the same phase as the other two, that suggests that the heterozygous parent had this gene order. We already had the phase from before, but now we know that the D gene is in the middle. So we have little e, big D, little f on one homologous chromosome, and big E, little d, big F on the other homologous chromosome. Okay? So now we have both the phase and we have the gene order. So the last step is to figure out the recombination fractions. Well, we have the phase here, so this is fairly straightforward. So we look and see which are recombinant relative to that parental phase. So let's start doing these in order. Let's compare D to E. So comparing gene E to gene D, the ones that are in red there are the recombinant ones, because you see they are not in the same phase as the parent. Because in the parent, little e is with big D and big E is with little d. In these ones that I've highlighted, big D and big E are together, or little d and little e are together. So when we total up those four numbers, it comes out to 36. There's 1,000 total individuals, so the recombination fraction here is 0.036, or 3.6%. Do the same thing for genes D and F. And again, we see the same sort of pattern here. Again, D and F in the parent are in the phase big D, little f, or little d, big F. So big D, big F, or little d, little f are now recombinant. So when we add those numbers up, we get 94. Okay, so the recombination fraction in this case would be 0.094, or 9.4%. We can stop there because that's probably good enough, but let's go ahead and take it one step further and look at between gene E and gene F. It should be close to, but not exactly, the sum of those two fractions. So we do that. Okay, in this case, genes little e and little f are together, and big E and big F are together in the parent. So recombinants would be the ones that are in opposite case. So these ones are all recombinant. Okay. And when we total up those numbers, it's 122. And again, that is close to, but slightly smaller than, the sum total of the distance from gene E to gene D plus from gene D to gene F. So at this point, we've solved the entire thing. We can actually draw it out. This is what we expect the parent looked like, so that we have the order, the phase, and the distances, or the recombination fractions, between each of those genes. So there we go. We've solved a more complex problem. I hope you didn't think that was too difficult. I know I did a little bit quickly, but feel free to go through the video again, pause, and work it through yourself. One thing I want to come back to, though, is looking at their overall procedure. First we identified and deleted the contributions from the homozygous parent, right? That was the first step. The second was to infer the phase of the alleles in the heterozygous parent, which again we use that by looking at the most abundant two genotypic classes. We then inferred the gene order in the heterozygous parent by looking at the least abundant two genotypic classes and comparing that to the phase of the parent. And the last one was determining the recombination fractions. So I want to bring things back and connect to the videos that are coming up by pointing this out. Why should you care? Like this cute kid here. Well, again, genetic mapping is used for localizing genes that affect traits of interest. It's not letters. There's no gene D, gene E, gene F. But this is the procedure that is done every day to localize genes causing human diseases, to localize traits of agricultural interest, for example. And yes, even though you have a genome sequence for many species, including humans, we don't know the function of all the bases. So we don't necessarily know where the gene of interest is. We don't necessarily know just by having a genome sequence, there's the spot that causes Alzheimer's when it mutates. And we also don't know the phase or order of genes automatically. This is the procedure by which genes are found. When people talk about finding genes influencing likelihood of getting diabetes or something like that, this is the kind of thing that happens. There are other procedures as well, which we'll go into shortly, but this is the type of procedure that happens. Well, I hope you found this helpful. I hope this was a useful bonus video for you. Thank you so much. Have a great day.
Hello, and welcome back to Introduction to Genetics and Evolution. Now we'll start tying in some of the things that we've talked about in previous videos about inheritance of multiple genes, so alleles of multiple genes at one time, with recombination and specifically crossing over to get to the principle of genetic mapping. How do we actually find genes related to diseases or other traits of interest? So let's get started. So what is genetic mapping? Well, mapping, simply put, is using associations between alleles at multiple spots in the genome. These spots in the genome are referred to as loci, to do one of two things. So either we determine where a gene is relative to other genes. So for example, in the case we were doing before with the Drosophila mutations, we were looking to see, where is the cinnabar eye color gene relative to the vestigial wing gene. Or we may be trying to localize a gene that is causing a phenotype. So we may be, for example, trying to find where is the gene that is related to diabetes predisposition or cancer predisposition. And this, again, is often done in the context of cancer. So this was one of the primary driving forces for the Human Genome Project a little bit over a decade ago, that we wanted to be able to map genetic diseases in the human genome once the sequence was obtained. Now, the goal from this was to basically find a way to find these genes faster and easier than could have been done in the past. So what they did is they essentially made this catalog of all 20,000 human genes. This was a huge undertaking because in order to do this, they had to assemble 3 billion bases of DNA into order. So just going along chromosome 1 and saying the sequence is A, T, T, A, G, A, C, C, C. That's actually exactly it. No, I'm just kidding. They essentially had to assemble this sequence along these long chromosomes. And again, it's 3 billion bases of DNA. So this was a massive international undertaking. But they were able to do it, and they were able to come up with the first draft human genome around 2001 or so. And they sorted these along the 23 pairs of chromosomes. This shows you what some of the chromosomes look like in a karyotype. Now, people thought, when you talk to the general public, they say, well, we have the whole human genome sequence. So don't you know where all the genes are that are associated with disease? Well, there's a problem there. And this is something I really want to emphasize because it's a common source of misunderstanding. It's not easy to go from DNA sequence to disease-causing gene because the DNA sequence looks something like this. A, T, G, G, C, G, A, G, C, T, A, G, etc., etc. It's not labeled, I cause cancer. It doesn't scream out to you that here's a gene that's important for this, or even is this necessarily a protein coding gene at all. So we can get some guesses or some hypotheses of normal functions of genes from the sequence, but we can only guess what sequences may cause problems. That's why we have to go through this somewhat arduous task of genetic mapping. Now, Genetic mapping is very much a first approach. It is not conclusive. Like you can't just map a disease and, oh, there it is, done, and say that's the base that causes it. It's definitely the start of looking into this. And what's happening when you're doing genetic mapping, this is very much like what happened with our Drosophila examples from previous lecture. We're identifying the location of disease mutations relative to other spots in the genome. These other spots are the genetic markers we talked about. So these are essentially reference points in the genome. And if you have a fully assembled genome sequence, then you know, for example, the reference point of this marker is at potentially base 1342 on chromosome 3, right? So that gives you a reference point. So along this, if I know it's this way from that marker, that way from that marker, I can localize and say, okay, it's between base 1342 and base 20,346,655. You can basically narrow down where in the genome these things are relative to genetic markers. And again, the principle is very similar to what we did with the Drosophila mutation. So how do we get genetic markers? Well, again, this is where we can leverage some of the resources that came about partly as a result of the human genome sequence. That multiple people were used for the first original human genome sequence, and we've had many, many, many more human genome sequences since that first one that came out around 2001. And what happened in doing this is you can compare nucleotide sites. You can basically put the sequences together and say, well, here's a stretch of sequence from one individual, here's a stretch of sequence from another individual, and look, there's a few bases that we see are different between individuals. You know, any pair of individuals will have a couple of base differences, as long as they're not necessarily identical twins, for example. So here we can say, well, this is a difference, this is a difference. We can use those as potential genetic markers. Now, these particular sites are referred to as single nucleotide polymorphism. Single, because it's one base. Nucleotide, because it is one of those bases of DNA. Polymorphism means a variation within the genome. So poly for many, morph for form. So single nucleotide polymorphism. This is often just abbreviated as SNPs. So it's a nice, catchy sort of term. Anyway, what we can do is we can, using blood or spit or something like that, and this is what they use for some of those DNA tests I talked about in an earlier video, we can genotype blood or tissue for these SNP markers and use them as reference points. So we can say that we know we can use this as a marker and then try to map a disease relative to this base. Use this as another marker and try to map the disease relative to that base, and maybe we can say it's between them or off to this side or off to that side, etc. So what resources do we have? Well, we have many. So first and foremost, we now have the genome sequence mostly put together from end to end on each chromosome in humans. Now, I'm emphasizing in this case humans. The same principle of genetic mapping can be applied to any organism. Now we have, in the case of humans, we have 23 long sequences corresponding to the chromosome. There are some areas that are not very well assembled, and there are some potential errors in there, but this is a very strong starting point. We also have computer-generated predictions for what parts are likely genes encoding for protein. Because again, the sequence is just like A, C, A, T, G, G, G. It doesn't tell you this part is relevant for making a protein, this part is not. But we can generate predictions, though, from computer algorithms for doing this. And last, but far from least, we have data showing some bases that differ between a lot of humans. We have this is referred to as the HapMap project, where we know a lot of particular sites in the genome, just like in the previous slide. Let me go back one. Just like in the previous slide, we have a lot of these mapped out, where you know some individuals have A and some individuals have G. So a lot of those are known, and we can leverage this last set as genetic markers for mapping diseases. Now, ultimately, the essence of genetic mapping is to look for an association between genotype or marker alleles and phenotype, or for example, a disease of interest. So if you tend to see an individual that has an A base at a site tends to have the disease, then it's likely that A base is either the disease-causing gene or at least very close to it, is linked to it, like we talked about last time. Okay? So again, this is just showing you what I just discussed a second ago. We have the genome sequence, just like this stretch of bases. We have predicted genes, and we have these HapMap SNPs, these single nucleotide polymorphisms. And these are the ones that we use as genetic markers. And these are, in essence, these differences between individuals are basically the same thing like what we were talking about with big B and little b. Like with Mendel's P's, those are caused by these sorts of differences. Now again, all genetic mapping comes down to seeing an association between genotype, the alleles at particular markers, and your phenotype, which is the trait of interest. I can't say that enough times, but that is what genetic mapping is. And that is how you are able to find along this whole stretch of sequence where are the genes that are associated with trait differences or disease predisposition. Now here's an extreme example. Let's say that we wanted to map a recessive genetic disease. Let's say it's sickle cell anemia, for example. Let's say that we're trying to map it relative to two genetic markers, A and B. Let's say at each of these genetic markers, they have two forms, big A and little a at the A locus, big B and little b at the B locus. Okay, so you've crossed two individuals. You don't actually cross humans, but let's just pretend. We crossed two individuals. One of them is a carrier for sickle cell anemia and is heterozygous for these markers. So this individual is big A, big B, little b. We also have this person have kids with somebody who has the disease, so may have sickle cell anemia. So little a, little b, little b in this case. Okay. Now let's say you get this set of offspring. So this first one is big A, big B, little b, and it is okay. Next one's big A, little b, and is diseased. Little a, big B, little b, and is okay. Little a, little b, and is diseased. So take a moment, look at this for just a few minutes here, and think about what would you conclude, whether the A marker or the B marker is closer to the disease mutation. Now remember, we don't assume that it is the disease mutation, we're just saying it's linked to it. One of these may be linked to it. So think about it, would it be the A marker, the B marker, both, or neither? Well, I hope that one wasn't too difficult. Again, the essence of all genetic mapping is looking for an association between genotype and phenotype. So let's look at the two genes in isolation here. Let's look at the A gene versus the B gene, and that's the easiest way to see what's going on. So when you're looking at the A gene, what's going on? When we have two possible genotypes. We have big A, little a, right? So the top two individuals are big A, little a, bottom two are little a, little a. Do we see any sort of consistent association? That if you're big A, little a, you tend to be diseased, or if you're little a, you tend not to be, or something like that? No. We see half the big A, little a's are okay, half of them are diseased, half the little a ones are okay, and half of them are diseased. So there is no association with A. So A is not associated with this genetic disease, or not clearly associated with it. What about B? Well, in this case, big B, little b's are okay, little b is diseased. Big B, little b is okay, little b is diseased. This very much suggests that B is linked to the disease. And specifically, the little b allele, individuals that are little b, tend to be the ones that get this disease. This would suggest that the little b allele is actually associated with some sort of disease-causing mutation in this example. So we'll elaborate on this quite a bit more in subsequent videos, but I hope you found this exercise useful. Thank you.